The primary memory function which is going on, and the cortex is a memory system, is sequence memory. Now I want you to think about this. It may not be obvious to you, but when you are trying to understand the world, for example, you're trying to understand my speech, the patterns are coming in time. It's like you're listening to a melody. The order in which the patterns occur, rapid changing patterns, is important. It's a sequence of patterns. The same is true when I touch things. And the same is true of my eyes. I'm constantly moving my eyes in the world, and there's a pattern, a sequence coming in. For you to understand or infer on the world, you have to have a memory of sequences. Similarly, if I'm going to make predictions, I have to have a memory of what follows next. You know, what follows this? What is typically after this? And that's a sequence memory. If I'm going to generate motor behavior, think about what happens to for me to create this speech right now. I am creating a very fast temporal pattern on dozens of muscles in my voice box that are going in this complex pattern. I am not making this up. I've said these words before. I've said sentences like this before. I'm doing little variations on a theme here and there. But basically, I'm playing back sequence memory of very complex sequences, and we do this in all our behavior. Sequence memory is key to understanding inference and prediction, and I believe this is the key to understanding how the hierarchy works. In fact, the idea basically is in the hierarchy is you learn sequences of sequences and sequences. You go up the hierarchy, you build stability. So let's just jump right into it here. We're going to do a little bit more neuroscience. Here's a picture of our brain again, a very light picture of our brain. We're going to zoom in on a little section of the neocortex. Hopefully you can see that better. There's a picture of a little slice of the neocortex. There you can see these layers going left to right. Those are layers of cells. And you can also, if you were close to this, you could see there's sort of a vertical orientation of columns in there as well. We're going to zoom in on one of those layers and look at a little caricature drawing that I've made here. Those little circles represent cells in the brain. I'm just making it so for illustrative purposes. And when you look at a little section of a layer of cells in your cortex, you see two principles. One principle is that, represented by the green arrow, is that there's a columnar structure. The cells that are vertically aligned in a very skinny column tend to have the same feed-forward response properties. They tend to behave the same way. Very few connections actually vertically, but there's a vertical thing. So if I give an input to the brain, those cells all have a similar feed-forward response property. The orange arrow basically represents where most of the connections occur. 95% of the connections are to cells and columns nearby. So we have a very vertical orientation in terms of performance. All these cells in the column are doing the same thing roughly, and then we have most of the connections are horizontal. If I now zoom in on one of those cells, this is that little picture of the neuron I showed you earlier, so we don't have to go through that again. But now I'm going to zoom in on one of the dendrites. And this is a picture of a real dendrite on a neuron. And if you can, I'll try to walk you through it. It's a little section. It's only probably in that picture maybe 40 microns wide or something like that. And you can see there's a branch going on in there, and you can see the actual synapses, the actual spines where the synapses connect. Those are the little appendages going up and down. They're really packed in about 1 micron apart. Now, these spines are all over, these synapses are all over the dendritic tree. We've learned something in recent years, which is very important, that these dendritic regions, like I take a little section of a dendrite, it acts in a very unusual way. When you have a bunch of synapses active at the same time, if I have one synapse active, it has almost no effect on the cell body. But if I have 10 or 20 of them active at the same time in the same location, it has a very large effect on the cell body. It's as if they're like a coincidence detector. It's like I have a whole bunch of patterns coming in at once. It says, oh, that's a whole group at once. That's good. If I only have some of them or a few of them, it doesn't do that. And so this is something that's a very important property, and we're going to need to exploit that in our model here. Just to give a little bit more flavor for that, since this is the neuroscience talk, so there's a little bit depth, and I hope I don't lose too many of you here. If I look at that neuron carefully, we can actually make a distinction between two types of dendrites. There's the ones that are very close to the cell body. These are called proximal dendrites. And if I have an input there, it has a linear effect on the cell, meaning if I have one spike come in and the cell gets depolarized a little bit, if I have two, it's twice as much, and three, and so on. So it's a linear summation. And this is where we find most of the feed-forward connections to the cell occurring. Then there are the distal dendrites. These are the ones that are further away from the cell body, and they work like I just said a little moment ago. There are dozens of these regions. They're nonlinear summation. They act sort of like coincidence detectors. And this is where we primarily find connections to other cells nearby. And these are important properties for us. We are going to model this with a model neuron. And this is a fairly sophisticated model neuron. This is a picture of the neurons we use at Numenta. And it basically models exactly what I just said there on the left. You have a cell body, which is that little square. We have the green dots are representing the proximal synapses, and then the blue guys arranged on those little dendritic segments are representing the distal synapses, and we will align them as a series of dendritic segments, not in a branch like we see in the brain, but in an array like that. And it has the same properties. So we're going to build networks of these guys, and we're going to learn how to form sparse distributed representations and learn sequences of them. So let's come back to this picture. We are now going to take that model neuron I just told you about, and we're going to arrange them in a layer of cells. And in this picture here in the center bottom, we're showing a lot of little rectangles, a lot of those cubes. Each one of those is one of our model neurons, if you will. And I'm showing them in an array like we'd find in the brain, and columns that are only 4 cells high in this particular case. And the colors of those cells are their activation states, which I'll talk about in a moment. Okay, so we want to understand is how does this structure, using those neurons, form sparse representations, and how does it learn sequences? And how does it make predictions? And how does it detect anomalies, and so on? Which is, I think, is what basically is going on in neural structure. Okay, so let's now zoom in on a layer of cells. You can think of this like the layer of bits in our sparse distributed representation, but this is a 2-dimensional layer of cells here. Each one of those cells is what I'm showing down here, and we have some feed-forward input coming from a sense or from another region in the brain going into this area. And that feed-forward input is going on the proximal dendrites, proximal synapses, if you will. And what we're showing in the colorization here, this is one of our simulations, the colorization here is representing the level of input activity that it's getting. It's not the firing rate of the cell, it's just the depolarization of the cell, how much input it's getting. Some are getting more, and some are getting less. They're sampling from the input space. And the ones that get the most fire first, and they inhibit the other guys. So, why did it stop working here? There we go. And so this little yellow thing is basically an inhibitory field, and basically the guys who get the most activity are the ones who inhibit everybody else. And what we have now is we have a sparse distributed representation of our input space. I won't go through how that is learned and how it exactly works because I want to get to the sequence memory. But this is how we ended up with this pattern. So here we have our array of cells. This is just like our zeros and ones. The white cells are zeros and the red cells are 1. Those are the active ones. I'm not showing you 2,000 columns here, about a quarter of that just to make it look visible. But you can see I have a sparse activation here. Now, over time, the patterns in the input change. I'm going to have to do this. Unfortunately, this is not working very well here. This is time 1, and okay, so here's time 2. So imagine now, as I'm speaking, as you're doing things, these patterns are changing constantly in these cells. Some cells are becoming active, and a moment later, other cells are becoming active as the patterns in the world change. And this is a sequence, and we want to learn the sequence. We want to learn, like, well, at this point, how do I predict what's going to happen next? And we do that on a cell-by-cell basis, and here's how. When a cell becomes active, it says, who is active recently nearby? Because if they were active recently nearby and I see them again, I'm going to predict my own activity. So a cell will come along and say, look, I just became active. Let me look around and sample from the people who are nearby who was active just a moment ago. And we're going to form connections to that. And we're going to do that on our distal dendrites down here. So this is like a coincidence detector. This is like our index of 10 patterns. We are going to basically say, I'm going to try to recognize the previous state by forming connections to it. And if I see that, I will become in a predictive state. I'll predict my own activity. So if I see this, I'll predict my own activity. Every cell is doing this all the time. And so when a new pattern comes in, we have all these yellow states, these cells in a predictive state. They're all saying, hey, I might become active next. I might become active next. Now, in this case, there's more yellow cells in predictive states than there are the red ones. And that's because, imagine I had some patterns. I had A followed by B, or I had A followed by C, or I had A followed by D. And if I show you A, it's going to predict B, C, and D. It's a union of those things. So that's what you're basically seeing happening going on here. And this is the basics of a transition memory or beginning of a sequence memory, because I'm learning how one state goes to another state. As I've shown it here, it has a major flaw. It's what we call a first order memory, meaning it only can go back one step in time. And that means I can't use information that occurred a long time ago. All I can use is what happened earlier. Well, let's say my patterns were XAB or YAC or ZAD. If I could go back 2 steps and say, oh, it's X followed by A, then I can predict it's B. If it was Y followed by A, I could predict it's C. And that would be a second order memory. We want a system that can go arbitrarily long sequences, learn very long melodies, if you will. And this system isn't going to do that. So the way we're going to solve that problem is we're going to add cells in a column. Essentially, we're going to use the columns and multiple cells to column to solve this problem. And let me show you how it's done. Variable order sequence memory. Let's start with our zeros and ones, our representation of a sparse distributed representation. And I'm now going to assign, instead of each of these bits being a 1, I'm going to make it a column of cells. So I now have 10 cells above it. So each column is now one of my 1s or zeros. And I'm going to pick one of those. Are supposed to be little circles above the 1s and zeros. I don't know if it looks like that to you. But anyway, I have 10 cells, and I'm just going to pick for every 1 bit, every column that's active, I'm going to pick 1 cell and say that's the one that's on. And I'm going to pick that for another 1 bit and another 1 bit, and I made an arbitrary assignment like this. I can now do the same thing again in a different representation. Here's the same representation, the same 1s and zeros, but I'm going to pick a different set of cells in that column. Now think about this. I now have the same input, but I'm representing in 2 different ways. At the same time, I can say they're the same input because they're the same columns, but on the other hand, I can say they're different because I use different cells. And even though some of the cells might be the same, 9 out of 10 will be different, and that's good enough. Think of it another way. If I have 40 active columns, or 40 bits that are 1, and I have 10 cells per column, there are 10 to the 40th ways to represent the same input. I have 10 to the 40th ways of representing an A, or 10 to the 40th ways of representing a pattern, a note and a melody, if you will. And I'll say a sentence here. I'm going to use the sound 2 in multiple ways. I'm going to say there are too many tutus to count. Right? Well, I use the same sound coming into your head over and over again, the 2 sound. But in context, your brain has to represent them differently because it hears them as differently, it perceives them as differently. And there has to be a way of doing that. And I propose the columnar structure like this is a perfect way of doing this. And it represents, gives us a huge capacity. So now we can go back and I'm not going to walk you through all the details of this, but if I do the same thing in a columnar array of a layer of cells, and a cell now makes connections to other cells in this larger assemblage here, we basically are going to build a high capacity sequence memory. Every cell is going to be predicting its next state, and when they do that, we can follow these and make predictions and go through time. I'm going to give you a little bit more flavor on it, a little bit deeper, so you can, if it gets hard, you can just, I'll come back to you in a second here, something easier. Part of this theory is that when a column is predicted or not predicted, it behaves differently. So if it wasn't predicted and a new input comes along and that column is selective, it fires all the cells. It's like, I don't know which cell is supposed to be active right now. It's like, I'm going to burst them all. They'll all become active. And this is a way like you can say, ooh, unexpected thing happened. Or it allows you to pick up a melody in the middle of the melody, something like that. If you had good predictions and you knew exactly what you're tracking and you knew exactly where you're going and everything was working just like you expected, then you'd only have activations of 1 cell per column. We see this kind of thing when unexpected things happen in the brain. You see a lot more activity going on. And so this picture here, I'm going to show you, there's 3 columns I'm going to highlight here. 2 of them, the 2 on the left, have 1 cell predicted, that's the yellow state, and the one on the right, which is a little harder to see, has no cells predicted. And if all those columns become active in the next state, then you'll see the one on the right, all the cells in the column become active, and the one on the left, only 1 cell become active. So we can detect anomalies on a column by column basis. It's sort of an attribute by attribute basis. Okay, so you put all this together in ways that you can read about if you want, and you end up with a very powerful sequence memory. It's a variable order sequence memory. It can learn very complex long temporal patterns. It makes multiple simultaneous predictions. It can detect when what actually happens is correct or not correct, and it can tell you why and why not. It is a high capacity memory. I'm not going to prove that to you here or demonstrate it today, but trust me that you can, a simple memory of just 2,000 columns can learn millions of transitions. It is a very high capacity system, and it's very kind of difficult to max it out. It's distributed. Think about this. There's not a single point of failure in this system. I can eliminate columns, I can eliminate cells, I can eliminate dendrites, I can eliminate multiple ones of those things, and it keeps on working. It degrades gracefully all the way down because of all the properties involved here. And finally, it does semantic generalization. What do I mean by that? Let's say I have a series of patterns coming in. And remember, these bits have semantic meaning. They're learned, but they have semantic meaning. And I learn some sequence of these patterns. And now I give you a new pattern. And it's the same sequence, but actually it's different elements. They're slightly different. Different numbers of, some of the bits are different. Some of the columns are different. It'll be able to say, you know what? These are different patterns, but I recognize the sequence. I recognize There are different spatial patterns coming in, different notes, if you will, but I recognize it's the same basic melody. And I will make predictions based on previous knowledge on a different environment. And so it generalizes in a semantic way, which is a very desirable attribute.